Hello there, it's Paras from Believers Academy. In short note series, in this video, we are going to start with photosynthesis in higher plants. Starting with definition of photosynthesis. Photosynthesis is an enzyme regulated anabolic process of manufacture of organic compounds. In simple, the process by which plant make its own food in the form of organic compounds using enzymes, carbon dioxide, water, and sunlight as a source of energy. As green plants make the food by photosynthesis by their own, they called as autotrophs, and all other living organisms are heterotrophs because all other are depend upon green plants for their food. What do we know is the balanced chemical equation of photosynthesis. 6 CO2 combined with 12 H2O inside chlorophyll in presence of enzymes and radiant energy, that is sunlight, which gives us one organic compound glucose, 6 molecules of water, and 6 molecules of O2 oxygen as a byproduct. Next is early experiments, but before this, in NCERT there is one more short experiment for starch formation. So first we see that. Okay, in this short experiment what we do, we take a green leaf and partially it is covered with paper and partially exposed to sunlight and air. After doing starch test, the exposed part shows positive results and the hide part shows negative result for starch. This clarifies that photosynthesis occurred only in green parts of the leaves and in the presence of sunlight. Next is moles half-leaf experiment. The same setup, but in half-leaf experiment, moles take one leaf in a test tube. Partially it is exposed to the air and partially it is inside cotton, which is KOH soaked cotton. So here is it. This is test tube. Half part is exposed to the air and half is dipped into the KOH soaked cotton. The exposed part shows positive results for starch and KOH dipped part shows negative results for starch. Why? Because KOH absorbs CO2. The portion of leaf that was in test tube don't get CO2 to make starch and tested negative. This shows that CO2, carbon dioxide, is required for photosynthesis and used as reactant to produce starch. Next is Joseph Priestley experiment, 1770s. That was too old. This experiment revealed the critical role of air in the growth of green plants. Okay, so Priestley takes one mouse and one candle and put them in closed bell jar. After some time, the mouse dies by the suffocation and candle also get exhaust. Then what he do? He again take one mice and candle, but in this case he add a mint plant in the setup and close it again with bell jar. After some time, the mice is still alive. Candle is continuous burning. How? Today we know that the mint plant released oxygen as a byproduct and mice and candle released CO2 by respiration and burning. But in 1770, Priestley doesn't know that which gas is released. So the air produced by burning of candle or animal respiration, he called them as phlogiston. This word is important. Phlogiston, which is converted into pure air or dephlogiston by green plants. Next is Sir Jan experiment. He experiment with aquatic plant that bright sunlight or sunlight have critical role for plant processes, purifying air by polluted air by burning of candle and breathing. When a bright sunlight is fall on green plant, small bubbles were formed, while in the dark such bubbles did not found. He found these bubbles to be oxygen. He found that the purification of air is carried out by green plants only in presence of sunlight. Next is in 1854, Julius von Sarge He proved that provided evidence for the production of glucose during plant growth. Plant produce glucose which is stored in form of starch, and starch is the first visible product of photosynthesis. Next is T. W. Engelmann. He used prism to split light into spectral components, then illuminating cladophora in suspension of aerobic bacteria. So this is prism, this is incident ray, white light, and this white light splits into the spectrum colors like blue, red, violet, yellow, green. What he observed that bacteria are accumulated in the region of blue and red light of split spectrum. This demonstrates that the effect of different wavelength of light on photosynthesis, and this is the first action spectrum of photosynthesis. Like these is the first action spectrum for photosynthesis. Yep, bacteria accumulate on blue and red light region spectrum, the first action spectrum of photosynthesis. Next is Carlinus van Neel. He discovered that photosynthesis, a light dependent process, involves reduction of carbon dioxide into carbohydrates by hydrogen form an oxidable compound. In simple, hydrogen form an oxidable compound reduces carbon dioxide into sugar, that is carbohydrate. So here's H2A plus CO2 in presence of light forms sugar, water, and free A. For further experiment, we have detailed topic, so we are covering them next. Now, where does photosynthesis take place? In green plants, mainly in mesophyll cells in the leaves, which have large number of chloroplasts. Usually, chloro- chloroplasts align themselves along the wall of mesophyll cells to get optimum quantity of light. This is the diagrammatic representation of chloroplast. We have outer membrane, inner membrane, then this is grana. These small structure you are seeing, these are not like plane or lines. These are in oval shape, stacked to form one grana. These the individual, it's called thylakoid, and when many thylakoids stacked on each other, they form grana. Then we have stomata, the outer inner space, then small ribosomes and lipid droplets, and starch granule. Next, membranous system, grana and stomal laminae. What is stomal laminae? These small bridges between two granas are called as stomal laminae, responsible for trapping of light, production of ATPs and NADPH, commonly called as light reaction. These are membranous system. Then stomata, what stomata do? Enzymatic reaction to synthesis of sugar is done in stomata, which in turn form starch take place. It depends on the product of light reaction, that is ATP and NADPH. By convention, it is called as dark reaction. This is only for conventional purpose. The process of formation of sugar might take place in presence of sunlight also, and it can be done at absence of sunlight also, because it is not directly depend on sunlight, it is dependent on product of light reaction, which are ATPs and NADPH. Next, how many pigments involved in photosynthesis? Pigments are substances that are have ability to absorb light at specific wavelength. We have four pigments. First is chlorophyll A, which is bright or blue green colored. Chlorophyll B, which is yellow green. Xanthophylls, which is yellow, and carotenoids, which is yellow to yellow orange in color. In maximum amount, chlorophyll A and chlorophyll B is present in leaf. There is another pigment. These are important accessory pigments for blue-green algae, which is cyanobacteria, cryptomonas, and red algae. Next is action spectrum and absorption spectrum. This is graph showing the absorption spectrum of chlorophyll A, B, and carotenoids. So these are wavelength in nanometers, nm. So you can see that chlorophyll A have maximum absorption in 400 and 700, which is blue light region and red light region. Chlorophyll B also have maximum in this region only. Carotenoids are absorbing light at 500 between 400 to 500 nanometers. This is action spectrum, rate of photosynthesis via the wavelengths. So we can see that it is maximum at 2 points in blue wavelength region and in red light region. If we combined the action spectrum to absorption spectrum of chlorophyll A, we can see that why chlorophyll A is primary pigment for photosynthesis, because it shows maximum absorption. It does maximum absorption. Now, what is light reaction? The photochemical phase involves light reaction such as absorption, water spreading, oxygen released, and the formation of high energy chemical intermediates, that is ATP and NADPH. The pigments are arranged into two distinct photochemical light harvesting complexes, that is HLHC, within photosystem 1, which absorb light maximum at 700 nanometers, and photosystem 2, which have 680 nanometer. So here's light harvesting complex. All the accessory pigments or pigments grabbing the light photon, that is light coming from sun, and passing to the reaction center. The reaction center is one of chlorophyll A, has a absorption peak at 700 nanometers. Hence we called it P700, or else PS2, reaction center PS2. It has maximum or maxima absorption at 680 nanometers. Hence we called it P680. Then the electron transport. First is non-cyclic photophosphorylation. First, photophosphorylation, the process by which ATP is synthesized by cells, is named as photophosphorylation. This is non-cyclic photophosphorylation. Lights. This includes both the LHC, light harvesting complexes, of P800 nanometers and P700 nanometers, both photosystems. Light falls on both the photosystem. First, starting with photosystem 2, by absorbing the radiant energy, it releases out its electron, which is grabbed by the electron acceptor, primary electron acceptor for photosynthesis, photosystem 2. Then it is transferred to electron transport system, and then goes to photosystem 1 P700. While this 1 ATP is formed from ADP plus 1 phosphate. Then after receiving the electron, this PS1 gives its own electron to its primary acceptor, electron acceptor, and by the energy of this, by taking this electron and grabbing H plus from stoma, NADP plus forms NADPH. This is non-cyclic photophosphorylation. Why? Because the electron released from the PS2 is not coming back to PS2. So energy PS2, first primary acceptor, then ETC, electron transport chain or system, then goes to PS1, then another electron acceptor, then NADP plus, which grabs one H plus and forms NADPH. The non-cyclic photophosphorylation is called Z scheme due to its shape. So if we see, here's the uphill direction movement of electron, then it goes down to PS1, then again one electron goes uphill to primary acceptor. So this movement of electron seems to be like Z shape. So that's why it is called as Z scheme. This Z scheme is proposed by Hell, Lee and Bendel. Okay, this is scientist name. They propose Z scheme of two photosystems. The cyclic photophosphorylation. This one is simple one. The incident ray or radiant energy fall on directly PS1, that is P700 photosystem. By this radiant energy, one electron is excited and gives to the electron acceptor, primary electron acceptor of PS1. Then it directly goes to electron transport system and comes back to PS1.